Okay, so today we're going to focus on single gene disorders. And the topics we'll cover in today's lecture are, I'll start with a historical background, and then we'll talk about patterns of inheritance of disorders among extended families, and then talk about specific types of genetic mutations that are known. And then finally, last week someone asked me about prenatal diagnosis, so I'll have a few slides about that. Okay. Now, we'll talk about four very important historical figures that really helped create the science of genetics at the beginning of the 20th century. Some of these you've probably heard of before. Okay, so I'm sure everyone knows about Gregor Mendel, who was a German-speaking Augustinian friar living in Silesia, which is now part of the Czech Republic. And in the mid-1800s, really all on his own, he started very extensive experiments in breeding plants. And you may have heard about this in high school and maybe imagined that he just had a small little garden and was making crosses. But this was actually a very extensive scientific program that he started all on his own with no funding or anything. And it's estimated that he planted about 30,000 plants during the time he was studying the genetics. So he did this in a very systematic way. He was trained in mathematics and he understood statistics, and he kept very meticulous records about the outcome of different crossing between plants. So this wasn't just, he was an amateur in one sense, but he carried out these experiments in a very systematic and scientific way and on a large scale. And through these breeding experiments, he was able to establish the rules of inheritance for discrete traits in plants. And this provided the foundation for the discovery of the gene and the establishment of modern science of genetics. Now, you must remember in the middle of the 1800s, there was no concept of the gene as such. And even Mendel didn't use the word gene. This concept came along later. But what he showed was that there were factors that determined the properties of plants. And these factors were something that were discrete and were hidden in the plant in some way. And they could appear in some generations and then disappear. But very significantly, they could reappear. And this went in later generations. And this went against the most widespread conception of what genetics was in the middle 1800s. And that was that people knew that family members resembled each other, and they knew that something was passed from generation to generation. But the concept then was that whatever this was, there was, whatever was present in the father and the mother was somehow mixed in the children. And then there was further mixing with every generation. And the new concept that Mendel's experiments gave rise to was the idea that that's not necessarily true, that there's something discrete and something that's non-changeable from generation to generation. It might be hidden in one generation, but then it can reappear generations later unchanged, essentially. And eventually that thing was eventually called a gene, and that's where the concept of gene originally came from. Now, by carrying out these very systematic studies, he was able to come up with two important laws, and those laws are stated here. The first is the law of segregation, and the idea here is that every individual, plants and presumably people as well, possess two factors. In the modern parlance, we call them alleles, but two factors for a given trait that separate from each other during the formation of gametes, during the formation of sperm and eggs. And they do this in such a manner that when the gametes come together again to make zygotes, these factors come together in an unchanged form and then can interact with each other. So this idea that these factors that we all have are somehow segregated and then come together is the first law. The second law has to do with what happens when you have when you look at separate traits and ask how they are related to each other. And the idea here is that they're independent of each other. So, for example, he looked at the smoothness of the pod or different aspects of the plant, and in his experiments and for the traits he chose, there was no relationship between these different traits. They're all independent of each other. And that's the second law, that traits don't influence each other. Now, nowadays, we know that the second law is not completely true, because we know that he discovered this is true because he happened to pick traits that were encoded by genes that are on different chromosomes or very far separated from each other on chromosomes. We now know that if traits are controlled by genes that are very close to each other in chromosomes, this is not necessarily true, that if you select for one trait, you may be co-selecting for another trait. But at least for the traits that he was looking at in plants, this second law held. Okay. Now, another person who was really contributed greatly to our early understanding of human genetics is Archibald Giroud. And he's a British physician who was active at the end of the 19th century and the beginning of the 20th, and was a professor at Oxford. And he was a clinician, and so he studied disorders in patients, and he focused on a set of 4 disorders. There's alkaptonuria, cystinuria, pentosuria, and albinism. And he was interested in these because these are disorders which they can appear in one generation, the disorder can appear in one generation, then disappear in several more generations, and then appear again. Furthermore, they're disorders that he noticed were common among children of marriages involving first or second cousins. So they were disorders that you saw at much higher frequency when there was a close blood sort of relationship between the husband and wife. Each disorder also, we now know, has in common the fact that it's a disorder, it's a recessive disorder that results when 2 essential enzymes are missing. And so in the case of phenylketonuria, let me just get here a little closer, it's missing an enzyme that's required to metabolize one of the metabolites of tyrosine, which is phenylalanine and tyrosine. And when that enzyme is missing, what happens is that this substance accumulates in the urine and then undergoes oxidation. And so when you look at urine that's been freshly voided, it looks just like regular urine. But if you let it sit for a day and let oxidation take place, this compound becomes colored. Either it becomes reddish or black, depending upon how much is in there. And so it was an attempt to understand this phenomenon that he began to look at families and to look to see how this was inherited. The other disorders he looked at are also result from the lack of an essential enzyme. So in this disorder, cystinuria, there are transporters that are missing that normally recover this amino acid, cysteine, and other amino acids from the urine and return it to the bloodstream. So when that's a way of conserving these very important nutrients. But when this doesn't happen, cysteine and other amino acids accumulate in the urine. And in the case of cysteine, this results in it precipitates and then eventually forms stones. And so the patients come with a very painful, a lot of pain in the abdomen and inability to urinate. And again, now we know it's because an essential enzyme is missing. Xylulose accumulates in the urine of people with pentosuria. And again, this is the result of a missing enzyme. And then albinism also results from the missing tyrosinase. And when this is missing, then the melanin is not produced, and you become have developed this condition of albinism. So what he discovered was he didn't know about genes at this time, and he only knew a little bit about enzymes. But he was looking at the pattern of how this appeared, these diseases appeared. He came up with a hypothesis that it was due to the lack of something that was essential. And furthermore, it was a lack that was present both in the father and the mother in a sort of a recessive manner. And then when it came together in the progeny of, first cousin marriages, both the lack from the father and the lack from the mother came together, and then that individual was missing something essential. Okay, and he noticed too that this pattern was very similar to what Mendel had found when he was looking at traits in plants. Mendel had also discovered positive, dominant traits and recessive traits in plants, and Giroud noticed that this also seemed to be the rule for human patients as well. So he was one of the first to make that connection. Okay, another important person is Thomas Hunt Morgan, and he didn't study patients, he studied fruit flies. But his group was the first person to really understand clearly the relationship between genes. By this time, the word gene had been invented, and he was the first to understand the relationship between genes and chromosomes. And by this time, it was also known that people had pairs of homologous chromosomes, and it became sort of there was a homology between Mendel's factors, which came in pairs and then separated and came together again, and the behavior of chromosomes, which we all have pairs. They separate during the formation of sperm and eggs and then come together again. And he understood that very well, and so understood that genes resided on the chromosomes, and was able to trace their movement through generations of breeding Drosophila. So his science, using the fruit flies, his experiments really established the modern science of genetics. Now, Fudan University has a connection with Thomas Hunt Morgan. You may have heard of Tan Jiajie, who is just recently passed away. He lived to almost 100 years old. And he was a very famous professor at Fudan University who established the first genetics department here on the Handan campus in China. And so he's considered to be the father of genetics. And he got his training in Morgan's group at Caltech early in the century, and then brought those techniques to China. Okay. Now, the last person I want to talk about in the historical introduction is Linus Pauling. And he's a polymath. He was just considered a genius in several different fields. His main training was as a quantum chemist, and he was one of the people who one of the first pioneers to take the new theory of quantum mechanics and show how it can be applied to chemistry. So he wrote a very famous volume on the chemical bond, for example. But later in his career, he became interested in proteins, their structure and function. And as part of that work, he analyzed the properties of hemoglobin isolated from normal individuals, and also isolated from individuals suffering from a disease called sickle cell anemia. This is a disorder that's very common, or fairly common, among Americans of African descent. And what we now know, this is due to a single base pair change in the beta chain the hemoglobin beta chain that results in one change of an amino acid. And what he found was that changed the electrophoretic mobility of the hemoglobin chain. And this is a figure from his very famous science paper that was published in '49. And what you're looking at is the profile of the migration of hemoglobin in an electrical field. And this marker supposed to mark the same position in each of these panels. So when hemoglobin from a normal individual is electrophoresed, the hemoglobin migrates here. When the hemoglobin from a patient with sickle cell anemia, however, is electrophoresed, you see the mobility is different. You get this is migrating faster than the other one. And if you have a person who's carrying the one copy of the mutant gene and one normal gene, you see a mixture. And then if you take this and mix it together, you also can see a mixture. Okay, so this very simple experiment showed for the first time that a genetic trait, which was already known to be a recessive genetic trait, could cause a change in a protein. And this was done before it was known that DNA was the seat of genes, that genes are made of DNA. And it was the first demonstration that a defect in a gene could not only result in the absence of that gene, it could change the property of a protein. Okay, so it showed for the first time that the gene had something very intimately to do with something tangible, a protein, and it changed the property of the protein. And because of that, this is considered a landmark discovery, and really essentially established the field of molecular genetics. Okay, so there are of course many other individuals who played important roles in the establishment of modern genetics, but these four are made particularly important contributions. Okay, so then the next thing I'd like to talk about are patterns of inheritance within pedigrees. Now, pedigrees just means an extended family. And what we're going to do is I'm going to give you examples of different types of inheritance that have been defined, and give examples of disorders that follow each of these patterns. Before we do that, though, I just want to show you, introduce a little bit about the notation here. In human genetics, we often talk about probands. And a proband is the person of interest. It's the person who comes to the clinic with a disorder. And so that person becomes the center of the study. And so the proband is marked here. And then in order to understand the genetics better, what doctors do is to make a family tree for that person. So here, these would be the brothers and sisters of this proband. This would be the father, this is the mother, and these are the grandparents on both sides. Okay, and this would be the children of the proband, and this is the grandson of the proband. So I think probably all of you have seen these kind of diagrams before. Is that right? Probably in high school. Yeah. And so again, the square represents a male. The circle is a female. Lines like this represent mating, and these are the offspring of the mating. This double line means a consanguous mating. So this would be a mating between cousins right here, first cousins. And to indicate that, we give a double line. The different colors here refer to the degree of separation from the proband. So first degree relatives, like brothers and sisters, are in light blue. So they're all one step away from the proband. And then second degree relatives are in gray here. And then there's some other notations that you'll sometimes see. Now, another sort of preliminary that we need to discuss are, first of all, what a genotype is. And what a genotype is a combination of alleles at a specific locus. So here, for example, is a picture of chromosome 1, and this represents the homologous pair of chromosome 1s that would be, that we all carry. And this band right here is, I've labeled with a big A or a little a. Now this band is much bigger than a gene. This is probably 50 to 100 million base pairs that you're looking at right here. But let's pretend that this represents a gene right here. So So one allele, one form of the gene would be on one chromosome, another form would be on the other chromosome. And in today's lecture, we're going to take the convention that the big capital letter is going to be the normal allele. It's going to be the most common allele in the population. And the little a is going to be the mutated or minor allele. Okay. Now, when we talk about genotypes, we also talk about whether they're homozygous or heterozygous. And homozygous genotypes just means that the person has 2 copies of big A or 2 copies of little a, whereas heterozygous individuals have 1 big A and 1 little a. And this could be big A might be from the father and this from the mother, or it could be the opposite combination. Now, to keep things straight, a very useful device is something called a Punnett square. And what this does is it lists the types of sperm or eggs that are available for mating here. So, for example, if the father is heterozygous at this locus, he would produce sperm that contains a large A allele and a small A allele separately and in equal amounts. And the mother, if she's heterozygous at that position as well, would produce eggs that carry either the big A or the little a. And then these come together randomly, and when they do, you get these combinations. And so there are 2 big A's here. There's one chance in 4 of getting this combination, 1 chance of 4 of getting this and this and this. Now, for a trait that is recessive, in other words, it only shows up if you have 2 mutant alleles, then you find that the offspring, 3 out of 4 of the offspring will be phenotypically normal, and 1 of the 4 will be have the disorder. And this 3-to-1 ratio is exactly what Mendel found when he was looking at certain recessive traits in plants. Okay, so if for disorders or alleles that that are dominant, where this is dominant to the mutated form, then depending upon what combinations of eggs and sperm, alleles in the eggs and sperm you get different ratios of affected or normal individuals. So, for example, if the father is heterozygous and the mother is homozygous for big A, then the combinations that you get are here. And if it's a dominantly inherited trait, then 1/2 of the offspring will, these 2 individuals, will have the disease, have the disorder, and these will be normal. If it's a mating between a male who's heterozygous and a female who's homozygous, 3 out of 4 would be affected and 1 will not be affected. So this is just the inverse of what you find for a recessive trait. Whereas any of the other combinations where you have, the father has 2 mutant alleles, or the mother's homozygous or heterozygous or homozygous for the mutant, all these other combinations, all the children would be affected. Okay, so then this is what we're going to do is match up then this pedigree analysis with the Punnett squares. And so, for example, if a de novo mutation appeared in this individual, now, of course, this was probably inherited from, well, it could actually be the mother or the father. If it's inherited from one of these individuals. Then the offspring here with a normal individual would be predicted to produce half affected and half unaffected progeny. And that's the same information as provided right here. Now, an example of this kind of, so an example of this kind of inheritance is a disease called Neurofibromatosis Type 1. And this is an autosomal disorder involving a gene on chromosome 17. Here's the frequency that's seen in the US population. The reason I said father, it turns out that 80% of the mutations are parental origin. And I'll explain why that is in a little bit later in the lecture. But here's what this looks like. One of the most common phenotypes is this so-called café au lait spot. You may have seen this in individuals where there'll be a discoloration of the skin. And this can appear anywhere in the body. And then these people also tend to develop subcutaneous of neurofibromas, which look like this. Now, some people are rather mildly affected like this, but others are much more severely affected. And if you look on the internet, you can find pictures of people who are just covered from head to toe with these kind of growths. It's really quite striking. Another sign are small nodules or tumors in the eye, in the iris of the eye. It's maybe hard to see this here, but they're little brown, small growths that occur within the iris. And then this boy has a distended eye here that's actually being pushed out from his eye socket by a tumor that's growing, that originated in the optic nerve that's growing behind the eye. And so as the tumor gets bigger, the eye gets literally pushed out of the socket. Okay, so what I wanted, so that's this very terrible disease. And this is a dominant disease. So if you have one copy of this mutation, you tend to develop this. When we think about these symptoms, there are two words that are important, or two characteristics that are important to know about. And one is the so-called penetrance of a phenotype. And penetrance refers to the idea that, suppose you have this mutation, and it's a dominant mutation. Even though you carry this mutation, you actually may not be affected at all for some reason. And whereas if you looked at 100 people, you may find 30 are not affected and 70 are affected. Okay? And in that case, you'd say the penetrance of this allele is 70%. And this is an all or nothing kind of judgment. In other words, it doesn't matter whether you're mildly affected or severely affected. If you're affected at all, that counts as being affected. Whereas if you're not affected at all, that counts as not being affected. So the penetrance has to do with the ratio of when you see the phenotype and when you don't see the phenotype at all. Okay. On the other hand, among people who express some of the phenotype, there's often a degree of severity. And so that's called expressivity. So people can have a very mild expressivity. So this person right here is more mildly affected than this person. And this difference is referred to as variable expressivity, not variable penetrance. This is all or nothing. This is a graded difference in phenotype. Okay, so the next pattern is the autosomal recessive inheritance. And here, what you see is, here are two probands that, since we know this is a recessive trait, they both must carry the mutated form of the gene. And from the fact that the parents are neither parent is affected, we can deduce that they must have this genotype, or it's possible that these are de novo mutations, but since there are two within one family, it's more likely to be inherited. And if you look at the Punnett squares, then this is what you see. So between two people who are heterozygous at this locus, you expect about 1/4 of the offspring to be affected. In a small family, this is 1/3 of the brothers and sisters. So that it's not, of course, in a small family going to come out to be exactly 1/4. But it sort of fits that pattern. Now, a famous example of a recessive disorder is cystic fibrosis. And this is an autosomal recessive. It's in a gene that it's called the transmembrane regulator gene. This is really a chloride ion channel, or actually a chloride and ion channel. And when it's defective, what happens is that the balance of water inside out of tissues goes out of balance. And it becomes, for example, in the lungs, instead of having normal secretions of mucus that you're able to clear by coughing, a very thick mucus develops within the lungs that the patient can't clear. And essentially the whole lung fills up with mucus. You can sort of see that. This is a dissected lung from an individual. It's just filled with goop. And of course, this becomes infected with bacteria, and just results in a miserable condition for the patient. And it's not only the lungs, but the intestines and endocrine glands, and in men, the production of sperm is also interfered with. So this is really a terrible disease. One interesting thing, we'll talk about this a little bit later, is that the frequency with which you find these mutations are very different among different populations. So, for example, among Northern Europeans, about 1 in 25 people carry the recessive allele for this, whereas in Asian populations, the concentration or the frequency is much lower. We'll talk a little bit more about this later and explain why this may be the case. Okay, so the other thing we'll talk about later too is that the expression of the symptoms is very highly variable. So some patients are very severely affected, whereas others are less severely affected. And this turns out to have reflect both environmental influences, but also the influences of other genes. And people have systematically looked for those genes to see how one gene can modulate the expression of another gene. Okay, so a good example of this is the disease called Xeroderma Pigmentosum. And this is a disease that involves the inability to repair DNA, and in particular, DNA that's been damaged by the sun. As you probably know, every time you go out into the sunlight, with some small probability, the UV component of the sunlight will cross-link the thymine residues in the DNA, cross-link them together. And when that happens, it creates a bulge in the DNA. And if it's not corrected, this by itself can give rise to mutations. So that there's a repair system in all of our cells that will cut out this cross-linked thymidine residues, and then replace the DNA with the correct sequence, like this. Well, these individuals are lacking a factor that is required for this process, and the net effect is this. It's very severe. So this is what happens when these children go out into the sunlight. So these children who have this disease are sometimes called children of the night because they have to stay indoors. They just cannot go outside, this is what happens. Now, in the United States and Europe, the prevalence of this mutation is very low. It's about 1 in a million. But in Japan, it's about 1 in 100,000. And maybe part of the reason is that in Japan, marriage between cousins is permitted. And so there are many more of these individuals than you'd expect just by random association of 1 in a million. 1 in a million. It's a small probability. Okay, yeah. The gray ones are where they didn't have information. They didn't have data for those areas. Yeah. Okay, so another pattern of inheritance is something is called codominant inheritance. And here you have a choice of where the choices are not, it's not, one is not dominant over the other, but rather you can have 2 different alleles and both can be expressed. And the good example of that is the ABO blood type. And the ABO blood type refers to the antigenicity of red blood cells. And that's determined by a substance on the surface of red blood cells called the H antigen. And this H antigen can appear on the surface by itself, and that results in the O blood type. Or it can be modified by the addition of an N-acetylgalactosamine residue. And if in that case it becomes an A blood type, or it can be modified by the addition of a D-galactosamine residue, in which case it's a B blood type. Now it turns out the enzyme that adds these sugars to the H antigen comes in two different forms. And they're two variants of the same enzyme, but one has a higher specificity for this sugar, and the other one for this one. And so if you carry alleles for the A, B, O, this would be, for example, the alleles that are carried in sperm here on this side, or this would be the alleles carried in eggs, and they can come together in these combinations. And A is dominant to O. So if you receive two A's, big A's, you're type A. Or if you receive one A and one O, you're type A. Or just the opposite here, type A. And the same is true for B. But A and B are codominant with each other, right? So if you're expressing both forms of this enzyme, they both are active. And so your red blood cells are covered with H antigen. And some of it has this attached and some of it has this attached. So this is an example of codominance. Okay. Now another example where it doesn't quite fit the dominance inheritance is incompletely dominant inheritance. And a good example of this is this achondroplasia, which is the most common form of dwarfism. And this is caused by a mutation in the receptor, the fibroblast growth factor receptor subtype 3 gene. And this is a mutation that does not inactivate this gene receptor, but rather makes it constitutively active. So it's always active. And we call that kind of mutation a gain of function mutation. I have that written down somewhere here. Here it is, gain of function mutation. And these are not uncommon among human disorders, where a mutation doesn't destroy something, but actually changes its property. And the incidence of this is listed here, and this is across all ethnic groups. And it's called incompletely dominant because it turns out only the heterozygous individuals survive. So this is a person who's heterozygote for this mutation. Individuals who are homozygote never survive. They're never born. It's lethal at the fetal age. So in this case, the phenotype of the homozygote person who's homozygous for these mutations is much more severe than the heterozygous, and because of that, it's called incompletely dominant. Okay. Now, so far we've been talking about autosomes, and the next few examples I'm going to talk about are for the sex chromosomes. So one. So the X chromosomes, again, of course, females have 2 copies, men have 1 copy. And so if you have a mutation on the X chromosome, this can be, since men only have 1 copy, if that is it destroys something that's essential for life, then men don't survive. And so this is, in this case, if a female carries this mutation, and if you look among the offspring, the males are all normal. And the reason that they're all normal is that any male that's inherited this mutation has already died as a fetus. So it's been aborted spontaneously as a fetus. But so only affected females are born. And then that can be passed on to the next generation. Okay. So and a good example of this is something called Rhett syndrome. And Rhett syndrome is a mutation in a protein that binds to the methylated CpG residues that we talked about last time. And if you remember from last lecture, methylation of the DNA is most often associated with the silencing of genes. It converts the chromatin from an open configuration to a closed configuration, and it does so by nucleating the formation of a complex of factors that all work together to shut down the chromatin. So what happens when you're missing this protein, you get inappropriate gene expression, and that is lethal. Now, for girls who are heterozygous for this, it's still it's still a very serious disorder, and this is a child with Rhett syndrome, and there are many symptoms, but the included are behavioral. There are cognitive problems and behavioral problems. There's epilepsy and significantly autism. This disorder accounts for a significant fraction of autistic girls. I'll just point out two characters. This girl walking on tippy toes here is one of the characteristic signs, and also this wringing of the hands that these girls often have this kind of position of their hands. And this extends even when they become old. All through their life, they sort of are doing this. And no one really knows why that's the case, but it's sort of a typical sign for these individuals. Now, this gene is also, the expressivity of this is also depends upon the pattern of X inactivation, which we talked about last time. So as you remember, women have 2 X chromosomes, but one of them is inactivated randomly. And so even though there's a 50-50 chance that one or the other will be inactivated, when that occurs, or how that occurs, this occurs in one cell during development, and then that cell produces progeny, and so you get a clone of cells where, say, the paternal chromosome, X chromosome, has been inactivated, and the maternal one is active. But another cell may have just the opposite pattern. The maternal X chromosome may be inactivated, and the paternal is still active, and that produces a clone of cells here where you have that pattern. So as I explained last week, women are mosaics. It's a mosaic of different patches of tissue where in some cases it's the paternal X chromosome that's inactivated. In other cells it's the maternal. And this balance is not necessarily 50%. And especially it's 50-50. And so depending upon, suppose the paternal X chromosome is the one that carries the mutation. Okay? Then if you're an individual where there's more paternal X chromosome that's been inactivated, then you're less severely affected by the disorder. Okay, because most of the cells are, or the majority of the cells are from the good X chromosome is present. So it depends on the pattern of X activation, how severe. The severity of the disorder depends on that pattern and the ratio of X inactivation.